There's a record button on the bottom.
Yes, I see it. Yeah, for some reason I was thinking it was in red, but it's not. So
Hmm.
I am going to go ahead and record now so that I don't forget, because I have a note here for myself and that still is probably not good enough.
Okay.
Um, okay, let's see. Admit. For everybody that's just coming on, welcome. And I've got you guys all on mute, just so you know. It'll just be Mark and myself that are going to be unmuted for the moment. Um, that's just to eliminate any extraneous noise in the background. So don't be alarmed if you can't hear yourself. I think Eve might be unmuted. And Eve, if you know how to mute yourself, I might get you to go ahead and mute. When we get done, if Mark still has time, we'll do some questions and answers at the end. If that's is that okay with you, Mark? Will you have a
It's
little?
fine. To tell you the truth, I don't mind if people want to ask questions during the talk because
Okay, sure,
that's fine with me.
sure.
If it gets too distracting, we can change it. But
Okay,
maybe let's just go ahead and ask while it's fresh in their minds.
sure. I'm a woman in my 50s, so any squirrel, bright shiny object, I'm gone. I'm gone. I'm gone. I'm hopefully, that's gonna fix itself here one of these days. I hope. We'll see.
Yeah.
Let's see. I still got people coming in.
Has anybody seen any unusual butterflies in Broward recently?
You know that they've seen the propona.
Oh, have they?
They have seen the propona.
At trade winds?
Yes, and lots of caterpillars. And we actually
Oh,
somebody found one in chrysalis. So that was
wow.
interesting. Yeah. So
That's fabulous.
and Barbara and I went back out there several weeks ago, didn't see it
Mm-hmm.
again, but we did, like I said, we found several of the caterpillars. Um,
Nice. On
yeah,
the
yeah.
Fertilary Shelter Trail there, or?
Yes, right there on that same trail.
No kidding. Wow.
Right there in that same location. And
Mm-hmm.
over by where the bridge is where they keep seeing the adults.
Yeah. Okay. Yeah.
Yeah, yeah. So very
Wow.
interesting.
One
Um,
of these days, I'd love to see it.
yeah, I know. Well, when one of these days when you can come back down, it seems to be from what I've seen,
Uh-huh.
we always go in the morning. It seems like later in the day is better, like
Oh,
after
really?
two, maybe 2 or 3 ish.
Okay.
So for what it's
Yeah. Good.
worth,
Good
yeah,
to know.
that might be a key
Mm-hmm.
to seeing it. And as for me, I have not seen anything. I have not been out. I'm trying to think if Barbara has reported anything interesting to me. She and I just did we did the Shao survey.
Oh, yeah. Did you see some?
She got to see one, and I did
Mhm.
not. We went in two separate groups.
Okay.
And, the mosquitoes at the very end, she and I got back together with her group, and they were going
Yeah.
on one of those trails that was just mosquito infested. And I said, I
Mhm.
think I'm going to sit this one out because they were biting me on my face.
They can be horrible
And
there.
yeah, and
Yeah.
of course, then she saw it. But I think the very first
Hmm.
day they went out, they reported that they saw at least one.
Okay. Uh-huh.
And have also found a guy out there with a net. They're hoping
Oh. Oh, okay.
not poaching, but they're
Hmm.
kind of keeping their eye out for that. So
Yeah. Right.
unfortunate, unfortunate. What in the world? Did people just want them for a collection? Why would I have no idea?
Yeah, I don't know. Or I don't know.
Yeah, I don't either. I mean, I can't imagine. You could sell that on eBay or
You
maybe.
end up in jail if you did, I think,
Yeah,
so
yeah, exactly.
yeah. Hmm.
Um, Okay, so I think let's see, it's 6:35. Let's go ahead and get started, you guys. So I am Melissa Lackey. Hi, everybody. Thanks for joining us tonight. I am the BCBC secretary. We have Peggy here. Barbara is away, and Cynthia has her hands full with some new babies and things going on at home. So I don't think they're going to be able to tune in. But we have with us tonight the infamous Marc Minno. He's going to give us a fabulous presentation on identifying butterflies. And we are going to skip the just the regular meeting portion, president's report, treasurer report, and we're just going to give you a nice presentation tonight. So thanks for everybody joining, and I am going to turn it over to Marc.
Thank you so much, Melissa. Thanks for inviting me and setting all this up. I know it's a lot of work to get all this together. And just as background, when I first moved to Florida, I had a job at the IFAS research facility there in Davie. And so Maria and I were there for, I think 3.5 years or so in Broward. And I did a lot of butterfly surveys at that time. So Broward, I'm still very interested in the butterflies of Broward and it's amazing what's still left there with all the development. You still have some of the rarest butterflies in Florida that occur in Broward County. They somehow hang on. But I'm going to start with my presentation. Let me see if I can share my screen. We're going to talk about how to identify butterflies. Can you see that?
Yep, I can see it.
Okay. And let's go to slideshow. Okay, good. So how to identify butterflies? Of course, butterflies are with moths. Here's a day-flying moth, the Nessus sphinx that you have in Broward. In fact, I may have taken that in Broward County. These form the order Lepidoptera, which are the scale-winged insects, as we'll see here. To talk about how to identify butterflies, you have to talk somewhat about classification because the science behind it is all based on the classification and the names and all. And really, our modern classification starts with Carl Linnaeus back in the 1700s. And he was mostly a botanist, but he was into everything. And he got it in his head that he was going to catalog all living things on the earth in the 1700s. And so he did exploration himself in Lapland and other places in Europe. But also this is the great age of exploration. So ships were going really all over the place at that time and bringing him back specimens from all over the world, which he named. And so he produced a number of these catalogs, Systema Naturae, and it's the 10th edition from 1758 is the one that's accepted as the beginning of our modern classification system. And Linnaeus also started with the classification system a method of naming plants and animals that's still used today. He came up with this hierarchical system. So from big groups to smaller groups to smaller groups, and he named them kind of based at the political systems of the time. You know, you had kings and queens and that kind of stuff. So he had the kingdoms. Back in his day, there were only a few kingdoms. There were plants and animals and maybe some other things. Today we have 7 different kingdoms. So things like bacteria or these ancient bacteria-like things, the archaea, protozoans, and plants, animals, fungi. So the kingdoms are the big groups. And then below that are smaller and smaller groups. So the phylum, the class, if we talk about butterflies, they're in the phylum, the Arthropoda, the jointed foot animals. They have jointed appendages. And the class is the Insecta. The order would be Lepidoptera. There are many different orders of insects, different groups, and butterflies and moths are in the Lepidoptera. And if we just take an example of a family, that's the Papilionidae. In Linnaeus's time, I think all the butterflies were he called Papilio, and that's still a current genus name, Papilio. And as an example, we have the tiger swallowtail. Here's an illustration. This is a redrawn one, but it's in Holland's book called the Butterfly Book. In the revised edition of his book, it shows the original illustration. It's believed to be the first picture of an American butterfly, and it was drawn in 1587. And this is what it looked like. And if you're familiar with a tiger swallowtail, you know, it doesn't have points on the tips of the wings and the tails aren't curved, but basically it's pretty much like that. But these days we have not only scientific names, but we have common names that the NABA, the North American Butterfly Association, and Jeffrey Glassberg have tried to standardize the common names for butterflies. And so their name for it is the Eastern tiger swallowtail. Now the scientific name, according to, and you may see this in books, the scientific name, you may see it as Papilio glaucus. And if there's a little L, an L next to it, that's the author's name, Linnaeus. He first described and named this American insect back in 1758. But there's been other classifications since his time, and you may see it in the genus Pterurus glaucus now. And if the genus has been changed from the original author, it'll be in parentheses. So you see his name's in parentheses, Linnaeus 1758 here. There could be other names associated with this butterfly too. Some examples of that. Here's a trinomial instead of a binomial. The trinomial means that there are different races or subspecies of that insect. And so the typical one would be Papilio glaucus glaucus. This is the common one all over eastern United States. But here in Florida, in peninsular Florida, we have a different subspecies. It's a larger, it's a deeper yellow color, a more orangey color. And so that name is used for the Florida one is either Papilio or Pterurus glaucus maynardi. You know, you may see some of these synonyms or invalid names too. There's a whole book of rules on how to apply these names. It's turning out actually that the common names are more standardized than many of the scientific names, because, you know, scientists are constantly looking at things and changing things. And so the scientific names have changed quite a bit from Linnaeus's time, but common names may be more standardized now. So how do you tell a butterfly one from another? And there's been all kinds of things. Basically, it's based on similarity, right? Is this one similar to another one? And they've looked at the morphology. This is the structure of the butterfly. So things like wing venation or color patterns, or the shapes or sizes of the antennae, the legs, the palpi, which are the sensory structures in front of the face, the genitalia. In insects, the genitalia, especially the male genitalia, are the gold standard for identifying species based on the morphology. And so we have some examples here. On the left here, I hope it's on your left, is one of the dusky wings. These are females. And so what they've done is they've snipped off the tip of the abdomen on a specimen, a museum specimen. They've digested away the soft tissues using sodium hydroxide. That's basically drain cleaner. And you're left with the exoskeleton and the hard parts. And so for the female, you see this plate here is shaped this way in this species on the left and this way on the right. And now there's variation in all of this stuff. And this is partly the confusing part of, to look in these details. There's going to be variation here, but most of the time this works pretty well and it's pretty easy. The only thing is, if you're out butterflying and looking at live specimens, you can't look at the genitalia on them, and you have to use the morphology, the color patterns and other things up here to tell them apart. Here's an example on the right of the males. The males have these clasping organs. Basically, they have to grab onto the female with the tip of their abdomen, and these have unusual shapes and things that are easily distinguished in the different species. But again, you have to be able to look at these under a microscope, and in museum specimens, that's what people do in museums. They basically look at this to help identify different species that are very similar looking. You can use other things too. You can use the eggs, the larvae, pupae. In fact, the first stage larva, just as it comes out of the egg, that first stage, the pattern of hairs on those it can be species specific. And again, you need a good microscope to be able to see that because they're very tiny, only a few millimeters. But there are whole books on these cetal patterns of the first stage larvae that you can look at and compare. So, but the problem in identifying species is variation. Here's a good example of this African swallowtail, Papilio dardanus. This is famous in biology for the variation. So you see on the top row is a typical male here on the left and a typical looking female. It's widely distributed in Africa, but in different parts of Africa, the females have different forms that mimic an unpalatable or unedible butterfly in a different family. So these are the models, we would say, over here. These are all related to monarchs. They're all in the milkweed family. So here's Danaeus chrysippus. Well, in one region of Africa, this Papilio dardanus, some of the females are, maybe all the females look like this, and look like the model, the toxic model. Here's a female of Papilio. Look at the resemblance. It's just an amazing example of plasticity in a species, and the variation from the typical one here. It's just astounding. How would you even know that these are the same butterfly? You have to raise them. The males always look like this. And so if you find a pair mating, you can tell, well, this one's hooked up with one of these, and it's like, oh, well, that must be the same thing. Or you raise them out and get males out that look like this. So so that's an amazing example.
That is amazing. Wow.
So we have classification, and people, have been doing classification for hundreds and hundreds of years. Classifying living organisms, but it was Linnaeus that really got our modern system going with his ranking system for the classification and his use of the scientific names, using mostly binomial, two names, the genus and species names. But then we have what's called systematics, and systematics came along with Charles Darwin. And here you see Charles Darwin as a young man and as an older man. He lived in the 1800s. He died in 1882. Of course, he's famous for his book and his other works. Since as a young man, he traveled the world on a ship to explore the world. And he saw many things that were quite different from his home in England. and it just got him wondering. He had lots of time on the ship to think about things and examine things. And also back home, he married a wealthy woman and didn't have to really work. He just, had a leisurely life and could pursue his interests. And so he wrote on the origin of species by means of natural selection in 1859. Just before this, another famous person at that time, Alfred Russel Wallace, Wallace traveled the Orient and other places of the world in the 1800s, collecting museum specimens that he would sell to finance his trips and make money. And he came up with the same idea, that this idea of natural selection is, everything is variable. All individuals are somewhat different from other individuals. Just like you could look at humans and the COVID thing going on right now. We're all different. Some of us get COVID and are perfectly fine. Maybe they don't even know you have it, and other people die. They get so sick from it. And so the variation is from our genes, our ancestry, and what combination of genes we have to fight it off. And so this is true of all living things. Butterflies, for example, a female butterfly may lay several hundred eggs, but probably most of those 100 or more eggs, only a few will be an adult butterfly again. Most of them are killed off by diseases or predators. And so this process of natural selection is winnowing out the population and leaving just the few individuals that can survive that particular climate or the particular predator or whatever's going on that is harming them. And so he wrote about this in his book, and then it became, we're not just classifying based on similarity for the fun of it, to figure out what's more similar or less similar. All of a sudden it became who's related to who, because his idea was that species split into other species and form new species sometimes. And so all of the species on Earth then, according to Darwin's idea here, came from some original ancestor long, long ago. And so the scientists then latched onto this idea and said, okay, well, now we have to classify things, but our pursuit is to find out who's related to who. What's the ancestry of this particular species of plant, a butterfly, a bird, or whatever. And so here's a copy of Darwin's book on the origin of species. And to take an example here, so now we're talking about evolution, change through time of species, and phylogeny, how are they related, and systematics, how can we classify them based on their ancestry. So here's a plate from one of Jeffrey, gosh, I just forgot his name.
Glassberg.
Not Glassberg, other
Okay.
Jeffrey, who does molecular looks at butterflies, Buckeye butterflies. But
That I can't help you with. Sorry.
it'll come to me in a minute, perhaps. Here, sorry, Jeffrey. So here we are in Florida. We have 3 different species. We have the common buckeye, Junonia senia. We also have this tropical buckeye. It's now called Junonia zonalis, and the mangrove buckeye, which is now called Junonia neildii. But then over in Texas, there are some other ones. Here's a different subspecies of the mangrove one over in Texas. There's this really dark one, and for decades, ever since I've been interested in butterflies, I've known about this very dark one, and nobody quite knew where to put it. Well, it's just recently been named as its own species. It's not a form of any of the other ones. And in California, look at this one with, it looks just like Xenia, just like the common buckeye, but it's a different species. And Jeff and other people have, Jeff Marcus, sorry, it just came to me. Jeff Marcus, he's in Canada doing research with the DNA and the molecular stuff. And just looking at these two, you would, and for years this went just as common buckeyes in California, and it's only through this looking at the molecules of them that we've determined that this is actually a completely different species in California. So amazing.
That's just crazy, crazy, crazy.
So butterfly relationships, people have been, ever since Darwin then, people have been drawing diagrams and trees and to illustrate how they're related. Here's a really early diagram. It's from Scudder, who's often thought of as the father of American Lepidoptera research. He wrote a 3-volume set back in the 1800s on the butterflies of New England, just packed with information. This is from another book, but here's his diagram of how the butterfly families fit together based on ancestry. And, you know, back in this time, in the 1800s, they didn't know really how they were related. They were just looking at them and saying, well, this one looks like that one, and this one looks like this one over here, or this one has this and that one has that. On his diagram at the top of the tree, which would be the most advanced forms, I guess, versus primitive forms down at the base of the tree, is number 1 are the satyrs. And then he has the milkweed butterflies and the heliconians over here, and I don't know, he has them arranged here in weird ways. And so that was fine early in the 1800s to kind of show the relationships here, but one of his branches at the very base here, he has, this is the skipper branch coming off the very base, the most primitive ones. And 13 here, this little branch here, he has equally branched with the other skippers. It's the Casneids. And these are actually moths. They're in tropical areas of South America, Central America. Here's an example from Australia. And they're very butterfly-like. They fly in the daytime. They have clubbed antennae. Here's an example of this Australian Casneid moth next to a giant skipper. And you know, you're right. I mean, those look really similar, you know, so no wonder he put them together. It turns out, though, that the Casneid moths are really far, far away from the butterflies in ancestry. And they're only looking like it out of, what we call convergence. So what do you look at to determine ancestry? Well, if you're working with birds or mammals or vertebrates, or shells, you're looking at fossils, right? And now it turns out there actually are some fossil butterflies, and Florissant, Colorado is the place in North America, at least, for fossil butterflies. This was an area millions of years ago where there were a series of volcanoes. It was a very tropical place at that time. And these volcanoes would erupt or explode, kind of like Mount St. Helens did in modern times in the West, and just cover everything with ash, volcanic ash. Everything died instantly, and it was pressed into these ash beds that became pressed into shale rock. And so there's been over a dozen, I think, butterfly fossils found in here. You find all kinds of plant fossils and other insects, flies, and whatever was out at that time, you can find in the rocks there. You can go there. It's a national monument and see their museum with some of the fossils. There are also private ranches there where you can go and dig up chunks of rock and look for fossils there if you want to pay them to do that. And it's a fun and really exciting thing to do. This is on the the west side of Pikes Peak if you get out to Colorado. And now this butterfly, I mean, we can see what it looked like. There's nothing left of the butterfly. This is just rock. But it was preserved in these shale beds in such a way that the color patterns even showed up. And now this is from several million years ago, and we don't know of any modern ancestor of this. Nothing in modern times looks like this. So, and I think that's true of all of the butterflies from Florissant. There's no modern equivalents, so they all went extinct at some point. And all we have are these few fragments in the rock to know that they were even there.
Mark, Arlene has a question. She said, are there butterfly fossils embedded in amber?
Yeah, there's a few of those. I just saw about a flower from amber from China. It's one of the oldest examples of a flowering plant. And so there's a lot of amber in the Dominican Republic that's mined there, and they often find insect fossils and other kinds of ancient critters that had gotten trapped in this tree sap that crystallized and turned into amber rock that's used for jewelry. Yeah, wherever there's amber, there's often insects that have gotten trapped or other things too. So it's hard to tell on some of them because they're all kind of gooed up together. You know, this was tree sap, sticky tree sap, and it's all kind of blobbed together. But some of them were quite elegant in there, and you know, in their preservation and everything, and you can see exactly what the insects look like.
Well, it's amazing that you can see the imprint and all the, you know, the color variations and the, you know.
We can see the venation, every detail of the antenna. I mean, it's just astounding.
It is astounding.
What's left, you know, of this impression. There's nothing left of the butterfly. It's just the impression in the rock. There are other fossils. There's some in Texas, and basically you just see an outline of a butterfly or maybe a few of the veins and things like that to be able to tell that what family it's in. I think there are butterfly fossils from Italy and Russia, other places of the world too. So we have some record, but it doesn't really help a whole lot. You may also see in journals or even online about, you know, how do we show these relationships? And there's a number of names that go with these phylograms or phenograms or cladograms. Basically, it's a series of boxes showing how closely related butterflies are. So in this case here, here's our Propona in the Charaxinae subfamily. And in this analysis, it's most closely related to the satyr, the wood nymph butterflies, the Saturninae. And then the lengths of these branches linking the boxes together shows how closely related they are, or how recently they've been separated in time. And so so you see these three are fairly recently derived, but these other ones are more distantly, way back in time, separated from them and diversified into these other groups. So I'm not really going to talk about, except that for each branch here has to have some characteristic of the butterfly that they have in common. So this Cyrostinae and the Nympholine here, there's one or more characteristics that are linking this box, this group together, that this group over here does not have. So ancestry versus convergence. Convergence is where different species of butterflies or other kinds of animals look similar, but they're not related. They're similar. Maybe they're in a similar habitat, and the selective pressures are similar on them. Maybe they're mimicking the same model to look alike. Let's look at this example. These are all are longwing or passion flower butterflies here on the left. And let me, it's been a while since I've given this. Let me look at this for a second. So so the top four are all the same species from different areas of South America. And look at the variation here. Again, how do you even know they're the same thing? Well, they know they're the same thing because they can look at the male genitalia on them and say that these are all Heliconius pneumata on top, even though they look very different. They're from different regions. And then these four below, these are examples of two other forms. This one's Heliconius melpomene, and this is Heliconius erato. And wherever these two species of longwings occur, they look the same. They tend to look the same. So even though they're different species, in this region, they would look like this. In a different region, they would look like this. And it's like, oh my God, you know, how do you tell these apart? And then, excuse me, on the right here, here's a plate I found on the internet of, I don't know how many, there's a dozen, these are all different species of butterflies that all have the same pattern, because they're all kind of, they're doing this mimicry thing where they're all looking the same, so that the predators don't have to learn different patterns. If they all look the same, they're all not worth eating. There's even a moth here. This is a day-flying moth that has the same color pattern in the same group. And so these are examples of convergence where different species have come to look similar based on selection pressures. And it has nothing to do with ancestry. So it's a huge problem of picking this apart. Are they similar because of ancestry, or are they similar because of selection pressures and convergence? Well, nowadays, we can use genetics and genomics. Genetics, we're looking at the genes and the chemical makeup of the genes. And if you're looking at many genes, they're calling us genomics. And we're getting to the point where they can do whole genomes of organisms to compare to other organisms. They've developed libraries of these DNA structures. So the big one that's used for classification is what's called barcoding. And they've taken one tiny piece that is called the cytochrome oxidase 1 gene. And it's a fragment of it. It's only 658 base pairs, I believe, of this gene, which has many more base pairs than that. And they're snipping that out. They're identifying exactly which nucleotides are in there, whether it's cytosine, adenine, thymine, or guanine. These are the four base pairs that make up DNA. And they're using not the DNA that's in our nucleus, but we also have another little organelle in each of our cells called mitochondria. The mitochondria are our powerhouses. They supply our energy in each cell, and there's lots of them in each cell. Mitochondria, they have their own DNA and do their own reproduction. And it's thought that this is from when cells first formed. The mitochondria were like a parasite or something that eventually ended up being part of the cell, because they have their own DNA separate from the nuclear DNA in each cell. So since the mitochondria are just dividing to make a new mitochondrion, they're rather than the nucleus in sexual reproduction, and sexual reproduction, the genetics are all shuffled and mixed up. So the mitochondria are a much more conservative way of looking at this. And they're looking at this one little fragment of DNA from the mitochondria and comparing it to all living organisms. So now we have a completely independent, well, not completely, but it's a different thing from morphology or the similarity we're used to compare things. And it's been amazing at in helping pick apart this convergence problem. This was one of the first big papers on this. So 10 species in one from DNA barcoding. This is the species, this beautiful skipper, Astrapes fulgurator, which is widely distributed in the American tropics, Central and South America. And at the time, this was 2004, it was only one species with some variation in there, but you know, it's widely distributed. So when they did the look at the DNA barcoding, it turned out to be 10 different species, not one. And then once they know that, it's like, oh, well, it's not just one, there's many things here. And then they start looking at the caterpillars. Look how different the caterpillars are. And they feed on different host plants. And then all of a sudden it's like, oh my gosh, yeah, that makes sense. That we had that information all along, but we didn't know how to interpret it. And now with the molecular stuff, we can interpret it. And look at this example from the internet. These things all look pretty much the same. There's 8 different species, 4 different genera, and 2 different subfamilies of skippers here in this group of skippers. And we only know that through the DNA analysis.
Mark, Arlene had another question. She asks, are mitochondria and blue-green algae related? That's a good question. Made him have to stop that one.
Well, let's see. Gosh, I don't know how to answer that. Yeah, I don't think they know exactly what ancestor the mitochondria came from. This was so many years ago. It's kind of lost. But everything that's alive has mitochondria. So this happened right when life came to be on the Earth. So it's not just butterflies that have been, with this knowledge now from the molecular analysis, the barcoding, and genomics, they're changing everything around. This is why the scientific names are changing so much. You know, they're changing from different genus names are changing around, different families are changing around, and it's true for plants too. There are huge changes in plant classification based on the molecular analysis. So here's an example of hackberry or sugarberry or Celtis laevigata. You have this in some places in Broward County. It's the host plant for 3 or 4 different kinds of butterflies there. Back in the day, it used to be put in its own family, the Celtidaceae. And then somebody determined, well, look at the leaves here. The leaves look a lot like elm leaves. And so they put it in the elm family, the Ulmaceae. And now with the genetic look, where did they put it? You'll never guess this. It's in the marijuana family, Cannabinaceae. You would never guess that this looks remotely like marijuana, but that's what the molecules are telling us.
If you see a bunch of people collecting hackberry leaves and rolling them into, you'll know why.
I can tell you they taste really good.
Oh yeah, the leaves or the berries?
The berries.
The berries.
The berries taste really good.
Good to know.
So now, not only can we use this molecular technique to tell who's related to who, but we can also use it as a clock, because the more different they are, the older they are, the more diverged they are. And so here's an analysis by Akito Kawahara and many other authors in 2019. Akito's here in the Florida Museum in Gainesville doing this genetic work. And And so you can't read this diagram, but we have the pictures that kind of show it. And so on the bottom here is a scale in millions of years, goes back from today at 0 to 300 million or more years ago. And so according to his analysis, the first moths to emerge were way before the dinosaurs at more than 300 million years ago. And we still have ancestors of these today. The tiny, I'm forgetting the name again now. They're tiny moths, only a few millimeters, and they have biting mouth parts instead of sucking mouth parts like most other moths have. And then if you look at the diagram, just stand back and look at it, you see divergences of other moth groups in here. But then late into the dinosaur period, at the end of the dinosaur period, look at what happened, this radiation of all these different groups, including the butterflies. And notice that the butterflies are coming out in the middle of the moth group here. And so this is what happened. Whatever caused the dinosaurs to die out, they think it was a comet or a meteorite that exploded in the atmosphere, and you know, everything that was big at that time couldn't make it. And then what do we have? We have our modern world emerging from that time. The dinosaurs are gone, all that kind of stuff's gone. Now we have flowering plants emerging, mammals, birds, and insects take off like crazy in association with the flowering plants. Just to show you some of the ideas on insects, how insects are related. So, you know, insects then have 6 legs, so they're called hexapods. That means 6 legs. But not everything with 6 legs are considered insects anymore. When I first went to college, we considered all of these things insects, but not anymore. These really more primitive ones, like the springtails and these soil-dwelling ones, the proturans and diplurans, these are tiny little things in the soil. They have 6 legs, but they're so different from the rest of the insects, they're not really insects. And same with these other groups. First insect groups are the mayflies coming off here, the ephemeroptera, and then the dragonflies. And then there's these branches coming over here with the grasshoppers. And here's a group here with the mantids and the roaches. If you look at a roach and a mantid, you go, yeah, my God, they look pretty similar. And that's because they're related. But then it also has the termites coming off of the cockroaches. Who would have ever thunk that? Termites look nothing like a cockroach, but the molecules are telling us that. And then over in this branch, we have way over here, the Lepidoptera, the butterflies and moths, together with the caddisflies, and a bunch of other different insect groups here. The bees and wasps, the beetles, neuropterans, flies up here, fleas, mecopterans. So that's kind of how the Lepidoptera fit into this grand scheme of all these different insect groups, just to show you. So when we get into this, the order Lepidoptera, which means scale-winged insects, if you look really closely here on the wings, here are the scales. These are these flattened hairs. They have a little base that fits into a socket that goes into the wing. Each scale has a particular color. So you see here white ones and black ones and blue ones, and patches of the scales then make up the various color patterns on the wings of the butterflies. The scales may be very tightly attached in things like monarchs, which are meant to be handled by birds. But other butterflies are very powdery, and that may give them advantage of being difficult to hold on to once a bird or something or a lizard gets a hold of them. They're just so slippery, they just slip right out of their mouth, and the scales break off. If you've ever grabbed a butterfly with your fingers, you know a lot of times the scales will stick to your fingers and there'll be a bare patch on the wing of the butterfly. Here's their close relatives, the caddisflies. The adults and the larvae have biting mouth parts in the caddisflies, and they are the hair-winged insects. Their wings are covered with little hairs, and you can see how hairy they are. But some of them are very colorful. They may have reds or other different colors, yellows mixed in, and are very moth-like looking, but they have biting mouth parts. So if you're identifying, now we get into our butterfly identification part here. So probably the best way to identify a butterfly is to look at the tip of the antenna. If you can see that, sometimes this is difficult to see, especially in the field when they're flying around, or even when they're landed. Sometimes it's not very clear, but if you can see that, in some of them it's very clear if you can get a good look at it. But butterflies, the butterfly antenna is thickened at the tip. We call this clubbed antennae. And so here's a typical butterfly. Now skippers are not only clubbed, but they have an extra extension called an apiculus coming off the tip. And often that's bent backward, or sometimes it's very small and hard to see. You may need like a microscope to even see it. So skippers are different from the true butterflies in the characteristic of their antenna. Now, some moths, these three over here are moth antennae. Some day-flying moths have, like we saw on that Castaic moth, do have clubbed antennae or something similar, but not quite the same as what the butterflies look like. Most of the moths have just a filament coming out like this. And it's the giant silk moths, though, that have the most amazing antennae, these giant feathery antennae. And here, if we're looking face on at a Luna moth, here are the big eyes on each side. The palpi are here. And coming off the top of the head then are these huge feathery antennae in the male. Females have feathery antennae too, but they're not as big as in the males. The males with these gigantic antennae are able to detect a few molecules of the female's pheromone up to 4 or more miles away. And as the wind's blowing these molecules to it, it's able to swim up that stream of smell and find the female very quickly. Amazing. All of the giant silk moths have these elaborate antennae. So where did butterflies come from? As I showed in that one diagram, they're showing up in the middle of the moth groups, and we think that their closest relatives, their closest living relatives, are inchworm moths, and specifically this group, the Heteleids. This is a tropical group. We don't have them in Florida or even in the US, I don't think. There's 1 or 2 in Cuba, and in Mexico and in South America, you'll find these Heteleid moths. They're just, the larvae look pretty much, they have some butterfly-like characteristics, and the adults are day flying with some color to it. You know, it's only because of the molecules that you can really tell that these are ancestors of the butterflies. Okay, I'm done talking about all that classification stuff, thank God, right? So does anybody have any questions about that stuff, though?
If anybody has any questions, just make sure you you unmute, or you can put them in the chat. You can type it in the chat, and I'll ask Mark. Mark, I have a question regarding what you were just saying about the scales on the butterflies. What is the let me go back to I put it on a note. Why would the monarchs be different? You said something about being handled by birds, I think.
Yeah, because monarchs are unpalatable, right? They're extracting the poisons from the plant, the milkweed plants, and they use that. They incorporate those cardinalides into their own bodies,
right,
to protect them from bird and lizard attacks. It doesn't work so much against spiders or invertebrate predators, but for birds and lizards, it can work pretty well.
Mm-hmm.
And so they don't know this, but they seem to, it's like they know that that the birds are not going to attack them. And they actually, with their flight patterns and everything, they're actually kind of inviting the birds to attack them. They're daring the birds to attack them. They have bright colors that are showing up very clearly. We call these warning colors in an unpalatable species.
Mhm.
And they have a very slow gliding flight, so any bird could pick them off very easily. Most butterflies fly very quickly because if they didn't, they'd get eaten by birds. Birds fly and catch them out, snatch them out of the air very easily. And so the bodies on monarchs are very rubbery. I mean, you can do a lot of handling on a monarch that would crush and kill a regular butterfly.
Mhm.
Wings, the scales are very tightly attached
Mhm.
because it's thought because they're meant to be handled by birds. So the young birds just coming out of the nest, they don't know one butterfly from the next, you know, or food item, and they have to try on their own. And they may go after a monarch and try it, and they can tell immediately that it's very bitter tasting and let it go. Or if they do eat it, they're going to throw up and get very sick and learn not to eat that.
So it's I mean, I'll say this a million times, nature is amazing, but that just is that
It's
boggles my
amazing.
that boggles my mind that,
So,
through natural selection, the
mhm,
monarch doesn't really need to have scales that come off because
yeah,
baby birds are going to say, "Oh, you don't taste so good," or get sick and die or whatever, and so therefore
yeah.
nature is amazing.
It is amazing. And it's not just monarchs that are like that. So all the unpalatable butterflies. So the a polydamas swallowtail, for instance,
Mhm.
highly unpalatable, very slow fluttering flight, um, tightly pressed scales. The zebra heliconian, another one, fluttering around, you know, daring birds to come after it.
That one's a slow one. If there was ever one that needed to be tasted, that would be a good one. Um,
Right. So even if you don't know, if you can tell just from their flight pattern or sometimes their colors too, whether they're unpalatable to birds or not, just from their behavior.
sulfurs are extra delicious, is what you're telling me. We have another, let's see, a couple of comments. Cynthia
Okay.
Waller said she's studying all the DNA and mRNA with her
Yeah.
grandson in biology. So this is
Oh,
great.
cool.
I hope her grandson's tuned in. And Arlene asked, have the insecticides worldwide changed some butterflies' genetic pattern?
Wow.
She's got good questions.
That's a good question. I mean, it could, because you know that's a selection process. If you're spraying DDT, for instance, all over a huge area, that's killing off butterflies that are susceptible. You know what happened with DDT though? You know, they were using it in crops against mosquitoes, and some of those insects survived and became resistant then. This is how natural selection works. You've killed off all the susceptible ones with that chemical, but some of them were genetically different and could withstand that chemical, and they were the ones left to reproduce and create the new populations that weren't affected by the chemical.
Wow.
So yeah, it's truly amazing.
So, along that same vein of questioning, how long do you think it will take before the monarchs become resilient to the OE parasite? I mean, do
I,
you think
I
that'll
think they already are.
are they?
I think they already are because we're finding from work from your group there, tagging monarchs and sending samples for the parasite, the OE parasite, into University of Georgia.
Hmm.
We're finding that South Florida and Central Florida, the monarchs are highly infected with OE.
They are.
But when you look at from the tagging program,
Hmm.
they live just about as long as any monarch does, and they're reproducing, they're laying eggs, even with this parasite. There's still monarchs down there. You all probably have them in your gardens, right?
Oh yeah.
I
Oh,
bet they're all highly infected with OE.
I just got a report back from University of Georgia that mine are all you know, I would say over 75% highly infected.
Yeah. Yes. Yes.
They're still surviving. You know, that's
And,
some good information for me to put out there on some of the Facebook pages, because people are getting so
mhm.
concerned and so wrapped up with bleaching their milkweed. And I would think
Don't do that.
yeah, no,
Don't.
the bleaching of the
It's
milkweed is going to cause even more problems.
Yeah, and it's just a waste of time. I mean, they're all infected anyway, or will
Yeah.
be
Yep.
infected.
Yep.
So
That's
why
good.
waste your time doing that?
That's a good reminder. I need to make sure I put a note out there for everybody. And
Yeah,
I told them what I passed along the information about the tropical milkweed as well, because
yeah,
a lot of people are scared to plant it. I said, hey, they need it. They need the milkweed, no
they
matter
need it.
what it is,
You're not going to have another milkweed down there that
and
can.
it makes them more toxic. That's why they
Yes,
gravitate towards that, towards the tropical, because it makes them
that's right.
toxic and less palatable. So, okay, great.
So, so with these monarchs that you have left in your gardens, those are the survivors. The ones that couldn't handle the OE died and are not reproducing anymore. Now it becomes a problem. I mean, if you're rearing monarchs inside in containers, that OE parasite can just decimate them. It's not the same. You have them crowded together in containers and inside. They don't get the sunshine maybe and everything. So that can wipe you out if you're trying to, farm butterflies. But in the wild, they're living with it and seem to be doing okay.
Wow. I hope that people will listen to this, because, there's
There's a lot of
a
misinformation
lot of
and just,
misinformation. Yep. And I
yeah,
try to
and old information out there that, yeah, you really have to kind of know what's going on.
yeah, yeah. And, invariably when I post things, I'll have 20 people want to argue with me. And I'm like, okay, it came right from Mark Minno, people. Okay, I'm sorry. I got off on a tangent there. Please continue.
Okay, I wanted to get into actually identifying butterflies. And, I turned 65 this year, so I had to sign up for Social Security and all that kind of stuff, Medicare. And so I started a long time ago, back in the 60s and 70s, and there just weren't a lot of resources available at that time, Uh, and nobody I knew of knew anything about butterflies. I learned it all from books, these few books. One of them I found in our school library, the butterfly book, written by William J. Holland. He was the curator at the Carnegie Museum in the 1800s. This book was first published in 1898. Look at the beautiful plates on it. I mean, amazing photography. And his philosophy was, we're going to put one of these books in every school library in America. And he did. At the time, there weren't that many schools in the 1800s. And so it was later revised, and I have a copy of this revised edition in 1931. This is the revised edition here. And I think you can still find it for sale. It might be kind of expensive, but and classification and everything is really old in it. But I learned, I would leaf through this book and these beautiful plates and wonder, where did he find all of these butterflies? Because around my house in Western Pennsylvania, I don't know, we had 20 or so different kinds of where were all these butterflies? It was amazing. And so that really got me going in butterflies, this particular book. Another amazing one was the Golden Guide to Butterflies and Moths. I don't know if you're familiar with the Golden Guide series.
Yes, I remember those.
Howard Zim, one of the guys that started this series, he had a property down in the Keys, and he would go down there in the summer times, I think. Because these are published out of Racine, Wisconsin. Amazing book. It features common and some rare things too, butterflies and moths. You see the beautiful illustrations showing how to, side-by-side illustrations and little text and wonderful range maps to show where they are in the world. This is just for North America, by the way. And I think it's one of the first books that actually shows the larva in nature, feeding on its host plant. And I could leaf through this and go, oh yeah, so that one's eating clover, or this one's on this other plant. And it has moths too, common moths. So this was $1. Can you believe it? $1.
Wow.
That beautiful work. And another one I found was the field guide to the butterflies, the Peterson field guide in the Peterson series. And again, this had more information in it and beautiful color plates. This is by Alexander Klotz. He was the curator at the American Museum in New York City, which was at the time, you know, competing with the Smithsonian to be the world's greatest museum. So those 3 books you can still find. This one has been revised and republished in a modern form by different authors. And so to identify butterflies, you need a field guide. You need something you can take in the field or come back at home with your photographs and leaf through. And the thing about Klotz's book here, the Peterson guides, based on the bird books that they did, they have marks showing what to look for on the wings. So these field marks here. So you look here or here, and he had a little on the explanation of the plate of what to look for too. So so that was very innovative to have these guides on the illustration itself, pointing to what you're supposed to look at. Nowadays, we have all kinds, we're swimming in information. This is the age of information, right? And so here are some websites. If you don't know about these, you might want to visit them. For taxonomic information, taxa are groups. The taxa are different groups. It could be a family, it could be a genus, it could be a species group. And taxonomy is the study of groups, of classification. So here's one run by the government, Integrated Taxonomic Information System, or ITIS. Now, if you just type itis in, that sounds like a disease, so you'll come up with some disease. You may have to type in some parts to it also to get this, but it gives you the complete taxonomic, all of the Linnaean classification from kingdom, phylum, all the different categories. It's all there if you ever need that. A wonderful one that people are using a lot lately is iNaturalist, and I think there's some other sites like this where you can just take a picture with your cell phone and send it into this site, and people there will look at it and send you an identification on it. And now sometimes they can't tell the species, but they can get it pretty darn close. And I think if 3 different knowledgeable people say that that's what it is, it goes into a higher category, and there are even scientists using these ones that they're pretty sure that's what it is for their, and they're finding some amazing things and using it in taxonomic and scientific studies from iNaturalist. Another one like it is Bug Guide, which has you know, just bugs, and you can send pictures of beetles or any kind of bug, flies. I was out with John Lampkin, and he took a picture of this tiny little gnat and sent it into him. And immediately somebody told him what it was. And that's what it was. It was amazing. I couldn't believe it. We also have Butterflies and Moths in North America site for butterflies and moths. They have range maps, pictures of caterpillars, and lots of living butterflies, and all kinds of information about the classification. And a similar group for a site for moths is this Moth Photographers Group. I don't know of any other moth site that has all of the information that this one has. Now, you may not be able to trust all of the identifications here, but this was started by a guy. He was a computer guy, and he started taking pictures of moths at his porch light. And he said, you know, why aren't people taking, all kinds of people are taking pictures like Jeff Glassberg of butterflies, but who's taking pictures of moths? And so he started this site, and it's just amazing. They have so many different photographs of so many different species. It's really a wonderful resource. And then for plants, I highly recommend the Atlas of Florida Plants site. If you don't know a plant, you can go here. You can send them pictures of it or specimens to them, or just leaf through the different photos there and try to identify your plants. It's a major resource for plants. And this is just for Florida, though. It's the USDA, the U.S. Department of Agriculture, has one it's for all of North America, so you can get an expanded view there.
Mark, before you continue, we had a question in the chat.
Oh, okay.
It is from, let's see, let me go back here for a second. Barbara Ritchie wanted to know if you could recommend a good source showing all the different instars of butterfly species.
Huh. Wow. There's just really scattered information with that. I know there's a Japanese book on swallowtails that has a lot of that. And there's just, I know our Southern Lepidoptera Society, some of you I think are members of that, there's some people that publish things like that. They take pictures of each of the egg, and each stage of the larva, the pupa, and publish studies in there with the photographs. But a book or some other source that has all that, I don't know of anything that's compiled all together like that.
Okay.
Daunting task, but it would be well worthwhile for somebody to do that.
And Arlene wants to know if you're going to help us with our butterfly count this weekend. Can you come down?
I wish I could. Sorry, I can't do it.
Rats.
Yeah, maybe it's some other point in the summer.
Maybe the summer count.
Yes. Let me try to finish up here. So I'm just going to go through some of our families at the family level. The family level is a good level to work on for both plants and insects. If you can put a family name on it for what you're seeing based on a field guide or just your knowledge or somebody telling you that's in this family, then you can do more research on it either online or with books and try to figure it out. So if you know it's a swallowtail, you can go to your section of the swallowtails in the book and try to figure this out and kind of know what to look for. I probably need to get moving here, but most of our swallowtails are black with yellow bands. They can be challenging to identify in the field, just flying around. And it takes some study to get familiar with them enough to really tell, say, a spicebush swallowtail from a Palamedes. You guys don't really have spicebush swallowtails down there, but up here in North Florida, you have all of these things flying around. It's like, what was that just flew by? And I've been out with people and they see a giant swallowtail and they go, oh, there goes a tiger swallowtail. And you go, what the hell are they looking at?
Yeah.
Because this looks so different from the tigers. But the underside of this giant swallowtail is bright yellow or pale yellow. And I can see what they mean. I mean, flying around, it's a yellow butterfly and you could confuse it for a tiger swallowtail. So some of them are so distinctive, like this zebra swallowtail, that there's nothing else really that looks like that here. Sulfur butterflies, they're tough. And when I go to Cuba, there's so many of them. They're just really difficult, and they have to sit. These little ones have to sit pretty much for you to see the spots on the underside or no spots on the underside, and they don't like to sit. So you can spend a lot of time just following one around, waiting for it to sit somewhere so you can identify it. If you're super familiar with them, you can probably tell. Or even flying, like this barred yellow. If you know what you're looking for, you can see this bar on it even when it's flying and know that that's a barred yellow. Gosh, well, we've been through the Statira sulfurs. I don't have a Statira on here, but the cloudless here looks like Statira. And then we have now the pink spot sulfur that we've recently, those pink spot sulfurs were probably in your area for decades, if not forever. And nobody knew they were there until Andy Warren just recently found them. And now they're they're one of the most common butterflies in Broward and in downtown Miami in the urban areas there,
Wow.
because
And
their host plant's a tree that's planted.
how long ago was that?
Andy did it. Well, maybe it's been about 10 years ago now that he was curating the specimens at the Florida Museum, and separating them and making sure they were identified correctly. And he found a couple of statira in a statira box from Miami that didn't look quite right. And he researched it, and he determined that they were the pink spot sulfur, which was only known from Cuba and maybe the Bahamas at the time. So he put it on his blog, because the specimens were from the 1960s from a school, collected at a school in Miami. And he put it on his blog. Has anybody seen this butterfly, or can you send me pictures of statira sulfurs from South Florida? And people started sending him pictures, and some of them were pink spot sulfurs. And then we determined what the host plant was, that Lysoloma sabacu. It's a tree from Cuba that's planted as a straight tree. And wherever that tree is, you can plant that in your garden, and they'll show up in Broward County, wherever you are. If you plant that tree, they'll come and find it. So we've only recently discovered that there's not just the statira, but also a close look-alike, the pink spot sulfur. So on these bigger sulfurs, you have to look at these lines, like here's the large orange with a straight line. So you have to compare the markings in your field guide to compare to the orange barred, which has a broken line here. Let's see what else here. Okay, hair streaks and blues. Wow, what to say here? So many of them, really, and they're so secretive because they're so small, and they may spend most of their time in tops of trees where you don't see them, or ones like Henry's elfin. You don't have this down where you are, but it's a pretty common butterfly in North Florida, but it's only out for a few weeks in the spring. So if you're not out there in those 2 weeks, you're going to miss it. Here's our carnivorous butterfly, the harvester. It's the only carnivorous butterfly in North America. The larva eats woolly aphids. It's a neat group. And then the blues. So again, a lot of confusion here into modern times between the Miami blue and the Knickerbean blue. Both of these butterflies are in Cuba too, but so to tell the difference, you have to count the basal spots. So on the Knickerbean blue, there's 1, 2, 3 spots here, whereas on the Miami blue, there's 1, 2, 3, 4 basal spots. So the Knickerbean is missing this spot. And it took decades for people to realize that these were 2 different species. And just to realize even that the Knickerbean was in Florida. We knew the Miami blue was here for a long time, but it's only been since the 1990s or so that we've realized that the Knickerbean blue was in the Keys. And I believe it's probably no longer there. I don't think anybody has seen it in recent times. I've had people call me up and say, I have Miami blues in my yard, in my front yard. And I go, okay, can you send me a picture? And it turns out to be the Serranus blue. And so you have to look. So here it just has 1 eye spot, whereas these 2 over here have the 2 eye spots, right? So again, you just careful looking and comparing with your field guides. We only have 1 metal mark in Florida, the little metal mark, beautiful little tiny butterfly, but in tropical America, these things go crazy. They're just so beautiful, and dozens and dozens and dozens of different species. Just a gorgeous group of butterflies there. I wish we had more of these in Florida. Then the brush-footed butterflies, we have so many of these. The passion flower butterflies. The brushfoots have really become specialized on just certain host plants, many of them. So we have the passion flower group, we have the croton feeding group, the leaf wings, we have the emperor butterflies on the hackberries, milkweed butterflies. Then we have some of these other groups, the admirals, Now, you don't have the red-spotted purple down here in Broward usually, but wherever. I get this one in my yard. It's one of the most beautiful butterflies, and it's so closely related. Here's a case of convergence where the viceroy has over time converged on the monarch pattern, and the red-spotted purple has converged. Let me go back here to the swallowtails, to the pipevine swallowtail, which is bluish black with spots. And some of these other weird butterflies, the American snout, there's a different snout butterfly in Cuba. I think we saw it with you, Melissa, there one time.
Yes.
And the ruddy daggerwing, they don't have this one in Cuba. They have two other daggerwings in Cuba. The brushfoots are a cool group. I really like them. So here's just some different examples. And then the wood-nymph butterflies can be quite challenging also. And for instance, here's a good example, the Carolina satyr. You know, for decades, we knew it as just the Carolina satyr. Well, a modern molecular study just a few years ago discovered that it's not just one species. There's two species in this. The intricate satyr they named as a separate species. It looks almost identical in color pattern. There are some differences, but they're very hard to tell apart, and they co-occur. I don't think you have the intricate one in South Florida, but it picks up in Central Florida and North Florida, and the two fly together in the same woods, the same sunspot may have the two species together. So, and then we have all the skippers, which are like the warblers of the butterfly world. There's many distinctive ones, but many that are very closely similar and difficult to tell apart, like the dusky wings. Where it takes some expertise and some study to really, and also the checkered skippers are that way too, of telling them apart.
That's a good comparison to say they're like warblers.
Here's an example of the of some of these dusky wings. So here's Horace's dusky wing on the left, and Juvenal's dusky wing. When you look at the top part of them, they're very similar. You can't go by the size of these white spots or anything. That's variable, and they have different seasonal forms. There's spring forms and later forms. What you have to look for on these is the underside of the hind wing right around in this area. So Horace's is missing these two whitish spots that Juvenal's has. And you see some pale spots here, but not distinct spots like these are. That's what you have to look for. I actually found juveniles in Broward County back when I was there in the 80s. One specimen I collected and never saw another one. It was up at the Executive Airport area with the sandpine scrub there. So I guess you could still have it, but likely it's probably disappeared along with the zebra swallowtail and those other scrub specialist butterflies that you used to have there in Broward. There's lots of different guides available. These are my guides, from butterfly gardening to caterpillars to butterflies through binoculars, butterflies of the keys, all kinds of quick identification guides to take in the field with you. And we do Cuba. We're exploring Cuba. We're finding new butterflies in Cuba. We published one just recently, a new wood nymph butterfly in Cuba. So if anybody's interested in going to Cuba, let me know. It may not be this year. It may have to wait till next year, but we're going to go again. We take jeeps up into the mountains where nobody else goes and find cool things.
It is an amazing experience. If you have the opportunity to go, I highly recommend it. You will learn amazing amounts of information.
I think it's the funnest thing we can do.
It's awesome. Yep.
Wasn't that fun?
It was. And I wish now that I knew more that I could go back, and maybe in a couple of years I will be able to go back with you.
The nice thing about Cuba is so many of the butterflies there are endemic, meaning found only in Cuba. probably 25 or 30% of the species there are found nowhere else in the world. And they have some of the smallest critters in the world. They have the smallest bird in the world, the bee hummingbird. They have tiny little skipper butterflies and other things like that are really small. So anyway, I'm done with the talk. I hope anybody's still awake.
Oh my gosh, everybody's awake.
Oh my gosh.
We had one person that just commented that one of the BCBC members found the pink spot
Oh yeah.
in Hollywood.
Yeah, it's out of Flamingo Gardens because they have the trees there. Anywhere there's one of those sabaku trees.
Oh, and we have
Sabaku.
added we've added Flamingo Gardens to the butterfly count.
You should see them there. Look for the tree. It has a shredding kind of bark to it, a flaking kind of bark.
What? Tell me again the name of the tree.
Lysoloma sabaku.
Oh boy, you're gonna
We
have
have a native Lysoloma but it doesn't have the flaking bark.
okay. Lysoloma. Okay.
And sabaku is a smaller tree, and it's in the legume family.
Okay.
If you plant one of those in your yard, you'll have pink spot sulfurs all the time.
Okay.
Roger Hammer showed me, he planted one in his yard in Homestead, and there were like hundreds of eggs on the new growth of his tree. It was unbelievable how many eggs they laid on this one little tree. And almost only a few caterpillars ever made it because the ants eat the eggs.
Oh, darn it. Yeah. There's that natural selection.
Yes.
Work. Yeah, we took a couple of trips down to the Everglades to go look for the Bartram's hair streak and the Florida leafwing, and did not
see
them. We did not see them, but coincidentally, I started emailing back and forth with Roger, and he told me just coincidentally that he and one of the biologists had found them like maybe a week after we had just been down there. So I told everybody.
You have to be just in the right spot at the right time.
Yes, exactly.
To see those. They've disappeared from everywhere else, and they're just, even in Everglades National Park, I'd say they're probably not doing well there either.
Yeah. I was glad to hear that he said that he saw them, but we did not.
Yeah.
We didn't see the pine.
Amazing.
Button, but no butterflies. So.
Yeah.
Let's see.
Now in Broward, I can say, you do have rare things there. So if you go over to the Ann Kolb Nature Center, you'll find the amethyst hair streak.
Yes.
That's coming out probably in May, in June is its time in the mangrove areas there.
Okay.
That's one of the rarest, but it took me decades to even see it.
That's good to know. I have never.
The amethyst hair streak. You have the tropical buckeye in Western Broward. You have the you have the other one, the silver banded hair streak out in some places.
Yes.
Broward too. You have the propona. Nobody else has a propona flying around.
We're special.
You are special.
I'm going to have to go see that amethyst hair streak because I have yet to see that. Now I just I got not a mangrove skipper, oh gosh, what's the other one that's very similar?
Hammock skipper.
Last year, my very
first
one on the count, and we had no idea what it was.
Okay.
I was thrilled, we couldn't figure out. We're like, what the heck is that? So yeah.
Now the amethyst we've learned from Paula Cannon down on Big Pine Key. She doesn't live there, and Amy and her partner there on Big Pine.
Oh yes.
Who have them in their backyard.
Yeah.
And so you can see them in the daytime sometimes on flowers or perched on vegetation, but it's not usually active an hour before sunset. And they're at the tops of the highest trees an hour before sunset. They're flying all over the place at the tops of the trees.
Okay. Well, that's good to know as well.
Who would ever think of looking for a butterfly before sunset?
Yeah, exactly. That's the crazy thing. And so many of us are looking eye level and down. We're not even paying attention to what's way up there.
Yes.
So good to know. I want to say they may have seen them at at Eula Johnson, which was I forget
Mhm.
what it was called before, the
They're in-
John Lloyd
Yeah,
Park.
I think they've been seen in even some people's gardens there in Broward County. And
Mhm.
they're at Hugh Taylor Birch sometimes.
Yes, yes, I think they saw him there as well.
Again, I think if you looked at just before sunset in some of those places, you'll probably see these tiny butterflies at the tops of some of those trees.
And when you said June?
May and June, I think, are its big flight time.
Okay. I'm making notes as we speak. I'm putting them in my phone so I don't lose them.
It's out at other times too, but that seems to be the time when most people see them.
Right. Okay. Okay, great. I don't know. Let's see. Nobody else has put any questions in the chat. Anybody else have any questions before we say goodnight? If you need to unmute yourself or put it in the chat or however. It seemed like I had another question, but maybe it was just about Roger Hammer.
Oh, he's
What a
working on all
character.
kinds of new books.
What a character he is.
Yeah, he's so knowledgeable, amazing. I've learned so much from Roger.
Yeah, we're hoping when he gets done with his books, he
Mhm.
may be able to speak to us. I'll make sure to let you know.
I'm sure he will.
In the
Yeah.
fall, yeah. He said he's crazy swamp busy right now, but we're trying to get him for the fall.
Mhm.
And we did get oh gosh, see, I'm doing what you're doing now. Ron McGill. We got Ron McGill for the fall. I don't know if you're familiar. He's the director of the Miami Zoo.
Oh yeah, yes. Okay.
Oh,
Yeah.
if you've never seen him speak, he is so animated and fun and so
Oh, cool.
passionate about nature, and
Mhm.
he's a lot of fun. So hopefully you'll be able to tune in for that one.
That's great. Yeah, I'll try to do that.
Mark, thank you so much as always. Your presentations just, I feel like my head is just swelling with all this knowledge.
Well, you're quite welcome, and thank you for inviting me. And
Absolutely.
catch up a little bit. And
Yeah, that would be great. And hopefully, maybe a trip down to South Florida, or maybe we'll come up to Gainesville and see you.
oh yeah, that'd be great. Come on up.
Yeah, that would
Love
be
that.
fun. That would be fun. We can go butterflying. Well,
Okay.
yeah. Well, thank you so much, Mark. And I'm sorry that Barbara wasn't able to be here, but I have recorded it, so we will send it to her, and she'll get to participate sort of, in another fashion.
Okay. Thank you so much, Melissa. Yeah.
Okay. All right. We'll see everybody soon. Everybody be safe. Take care. And I'll see some of you on Saturday for the count.
Okay. Bye-bye.
Bye. Take care.